First, I'll talk about it as a clinical efficacy biomarker. Obviously, the FDA won't accept something like electrophysiology because doesn't change patients' experience, but it certainly can give you a lot of information on, is your drug having a positive effect on the retina? Then I'll talk about its use in safety assessments. And then finally, the translational biomarker going from animals to humans to inform your first-in-human studies. So as I was saying, the RETeval is really, in my mind, enabling technology for multi-center clinical trials. It's non-invasive, so it uses skin electrodes. You don't have to put contact lenses in patients. For the diabetic retinopathy protocol that they have, it's all photopic, so you don't have to dark adapt. It's easy to customize protocols, so you don't have to have the technician make mistakes. They put in the protocol for your study, and it basically runs it itself. So it's like a turnkey type system. And yeah, the level of expertise for the technicians is you don't have to have a PhD to do it. The device meets ICEV standards if you want to do, ICEV standard electrophysiology. It's validated against more conventional technologies using contact lenses and requiring pupil dilation. I forgot to mention that, this has two modes. You can do it in the way you normally do with dilating the pupil, as normal ERGs are done, or you can do it with normal pupil, and it'll estimate the pupil diameter and adjust the light intensity. So you're controlling how much light gets back to the back of the eye. So, when people, usually when you talk to retina doctors about doing an ERG, they'll groan and say, we don't want to do that. And I think the RETeval really, should change that in terms of the burden on the site, the burden on the patient is really minimal.
And you can use it in pediatrics as well?
Yes, for sure. Quentin actually just made a presentation. They design new electrodes that are smaller, so it fits on a small child. So, and they're busy validating that now. So yeah, and the big thing for small companies is, you don't break the bank to buy this for multi-sites. It's inexpensive equipment. This is a study that I did with Quentin that I think demonstrates the use of the ERG over, in addition to more standard, imaging techniques. And, there are myriad of publications saying, look, here's the ERG, and it gets worse as, say, diabetic retinopathy gets worse. But, clinicians are used to doing diabetic, looking at the back of the eye and seeing diabetic retinopathy. They're not used to doing ERGs. So why are they going to do an ERG? They don't know how to interpret it. So Quentin and I decided to look at how the two work together. So if you have, in this case, we were looking at diabetics, and we had the diabetic retinopathy severity score, the standard on fundus photograph. So what we did was they had, when they first validated their flicker ERG protocol for diabetic retinopathy, they took a bunch of patients with different stages of diabetic retinopathy, different grades, and they measured the flicker ERG. And what we decided to do here was follow up those patients for up to 3 years. Okay, so 3 years after the study, after they had these initial, we looked at how many patients had to be treated with anti-VEGF for diabetic macular edema, or had to be treated with laser for proliferative disease. So the intervention was intervention for progression of diabetic retinopathy to where they needed treatment. So what we're looking at here is, these are patients that had, we dichotomized the diabetic retinopathy severity score. So eyes at high risk had severe non-proliferative diabetic retinopathy or worse. So they had, or proliferative disease. They're at high, we know from the original ETDRS study, that that they're at high risk to progress. And that's what makes the DRSS score an accepted surrogate, was that data from the original ETDRS study way back when that shows the prognostic value of having an eye at risk with a high DRSS score. So we looked at patients with low risk and patients at high risk. And we saw how the ERG, the ERG flicker score, so adds to the information that you get from the fundus photographs, okay? So you can see, let's look at the 2-year data here. These are patients that had ETDRS less than 53. If their diabetic retinopathy score was lower than 23.5 criterion, their risk was low. If their score was greater than the criterion, their risk was higher. Okay, so the ERG has added value in these low-risk patients. This is the mean of the patients that had an ETDRS positive score. So they had eyes at high risk just based on ETDRS. And you can see the risk of having an intervention in these, quote, low-risk patients, if the ERG was positive, was approximately equal to that of those, quote, high-risk patients. So the ERG added a whole lot of prognostic value. And if you have a patient that you wouldn't follow closely because their retina looked okay, if you added the ERG in there, their ERG looked poor, you should follow those patients more carefully because they're at higher risk. And the same is true for these patients at high risk. I mean, by ETDRS, these are high-risk patients, but look what happens if their ERG is lower than criterion. Their risk goes down to less than 10% intervention at 2 years out. So less than 10% of patients actually progress to needing treatment for their diabetic retinopathy. And that was equivalent to if their ETDRS was negative, so less than 53. So what you get, so this patient where you might be very worried and think, well, I'm going to start them on anti-VEGF because their retinas look at high risk. If your ERG is normal, you don't want to treat that, and you don't have to see them very. So what we demonstrated here was how powerful adding function to structure is in prognosis for patients. And in addition, if you're doing a clinical trial and you want to enrich your diabetic population, if you take ETDRS patients that are positive and they have a very abnormal ERG, you can really enrich probability that they're going to progress over the next 2 years or 1 year even. So I think this paper I'm proud of it because I mean, it really shows the value in a way that should be meaningful to clinicians in terms of how to manage their patients. And it's a value also in clinical trials for how to enrich your populations.
The one coming down is for the physician, the one coming up is for us, pharma.
That's right. Exactly.
So has FDA or other regulatory bodies, are they aligned with this, and is this potentially useful as an outcome or an endpoint? That
I think it will. I mean,
at least the bottom line for us.
Yeah.
If the regulators.
Yeah. Well, I don't think the regulators are very interested in patient experience, so the ERG doesn't meet the surrogate, in terms of.
So what about clinicians?
Yeah. Well,
are they adopting this as a kind of a risk assessment?
I mean, hopefully. So I'm on now a juvenile diabetic retinopathy foundation. We're trying to redefine progression of diabetic retinopathy because, I mean, the ETDRS is just based on vascular appearance. And it's become very clear that there's a neurodegeneration that occurs in diabetic retinopathy that's independent of how the vessels look. And the ERG can inform that. Yeah.
What's the definition of PLS-VR positive? What's the definition?
Yeah, so they had a score of 53 on the diabetic retinopathy severity score. So it's severe non-proliferative diabetic retinopathy or worse. So they had either.
So it's not the number of letters.
No. It's the scale. So the DRSS score is the scale that's used by regulators. So what's now used for approval for drugs for diabetic retinopathy is a two-step improvement in the score, but it's completely based on hemorrhages, you know, all the vascular changes that you see.
It is somewhat arbitrary based on that particular study because it depends on blood pressure. They convince and work with the regulatory agency to put it into those two-step improvement as a benchmark, because it's very hard to really gauge how the progression of the diabetic.
Yeah, so I mean, but what payers will pay for though is preventing ever having to start anti-VEGF or ever having to get laser or ever losing vision to diabetic retinopathy. So it's accepted as a surrogate, but the real outcome is actually like the intervention. Yeah. Just in general, I think this strategy should be used to combine information. You know, how much dysfunction add to structural information. So based on this study, actually in the ARPO study that we did on diabetic retinopathy, we put the flicker ERG into that study. You know, we were recruiting patients that had fairly normal vision. So they had moderate to severe non-proliferative diabetic retinopathy, visual acuity is 20/30 or better. So they had good vision. So visual acuity is not going to be your functional endpoint. We were looking for change in diabetic retinopathy severity score, but as a functional, I was hoping to see that the treatment would actually improve the ERG, the flicker ERG. So it was, you know, a typical diabetic retinopathy study. We recruited, I forget how many patients, but patients with moderate to severe non-proliferative diabetic retinopathy, no DME, good visual acuity, and they were randomized there. So we had 167 patients randomized either to receive drug or placebo. Okay, and it was a one-year study in duration. Unfortunately, the drug didn't work. So we had done a phase 2 study, an earlier phase, the DME study where we, a 3-month study where we showed about a 10% of patients had a reduction in their diabetic retinopathy severity score. And we thought that if we went for a whole year, we'd see a lot more patients. And that this was it was a systemically delivered drug, so patients could dose themselves at home, and we thought it could be used as a preventative drug. Unfortunately, in the year-long study, we still only had 10% responders. So it's hard to do a preventive treatment that only treats 10% of your population. So we canned the drug, but we used the ERG to replicate what we saw in the first study. You know, how does its structure and function work together? And we collected also OCTA data, fluorescein angiography, ultra-wide field data, in addition to the ERG. And we found again, so this is the number of patients that had each of these tests longitudinally. I won't go into all the details of the study, but we found again that the ERG was the most predictive of who was going to progress, either get 2 steps worse on the diabetic retinopathy severity score. So I won't go through all the methodologic details, but as I said, we got ERGs, we got OCT angiography, we got wide field fluorescein angiography. And we quantified a number of parameters on each of these. And interestingly, despite what Jocelyn's been saying, we didn't find ultra wide field fluorescein angiography to be useful at all in predicting progression. But again, we found that patients that had a high score on the RETeval were, so this is plotting score of the RETeval on the horizontal axis, and then the probability of progression on the vertical axis. And you can see the patients that had high RETeval scores were much more likely to progress than those that had lower RETeval scores. And if you just looked at the implicit time of the flicker response.
Was this compared to FA?
No, this is just,
The FA didn't have any statistics.
I don't know, I can't get it to
But
move
it's not.
now.
It was.
Yeah. So the OCTA, the foveal avascular zone expansion did show some prognostic value that was statistically significant, but you can see the separation of the ERG in negative versus positive was much larger than the OCTA on its own. And we have some data showing that combining the two together, just like we did with the DRSS, is more informative than either one alone. So basically, we in a prospective study, we actually validated what we had done in the retrospective study, showing that combining structure and function is better than either one alone, and that in this case the ERG was definitely more sensitive to prognosis than fluorescein angiography or OCTA. So
How long was the prospective study? Sorry.
1 year, yeah. So this is looking at change over a year. And the events we were looking at was what defined progression was two-step worsening of the diabetic retinopathy severity score or development of macular edema.
So if you made the sensitivity difference between the score and then your actual ERG result, you say that it's a double. How do you?
Yeah, I think in this study, I mean, the score did better than just the ERG alone. It also looks at the pupillary response. It combines, so it's kind of an intelligent algorithm that LKC defined. But just looking at the flicker implicit time is very good and did better than any of the structural measures. So I'm not sure that we quantified how much you gained by score.
The answer was, so if your DR score was greater than 26, you had a 41% chance of progression in the year. And if your score was less than that value, you had a 4% chance of progression. So there was a relative risk change of 10.
The photopic negative response, I thought I'd just briefly talk about that. This is useful in optic nerve and retinal ganglion cell diseases. It's been used a lot in glaucoma. So photopic negative is negative peak that occurs after the B wave. And in Laura Frischman in Houston did a lot of work to validate this. In this case, she treated monkeys with tetrodotoxin, which blocks all spiking cells in the retina. So you inject it in this, and so ganglion cells and amacrine cells just aren't responsive anymore to light. And so you can see that here's a monkey before treatment. After treatment with tetrodotoxin, the A wave and B wave remain unchanged, but this negative response is wiped out. So it shows that it's generated by the ganglion cells. And they've published, there's been a whole lot of publications in glaucoma patients and patients with optic neuritis and various optic nerve diseases showing that this photopic negative response disappears or it becomes very abnormal. Quentin's been doing some work on how to optimize the measurement of it, so to get more reproducible data. There's a lot of things you should do, because if you get drift, you know, if your baseline's drifting up or down, you can get all kinds of artifactual measurements. So this is looking longitudinally at a patient over 4 years, and here's a glaucoma patient. And you can see their visual field progresses over those 4 years quite a bit. And this is looking at their nerve fiber layer. You can see there's thinning of the nerve fiber layer. And you can see the ERG after year 3 becomes substantially photopic negative, disappears, and kind of remains disappeared over the next year. So a lot of the electrophysiology is sensitive to early in optic nerve disease. I think that's with the pattern ERG, the same thing has been shown. His dynamic range is best early in disease, and for late disease, it kind of washes out before the fields do. So, but certainly if you're treating early in glaucoma, electrophysiology should have a good sensitivity specificity. Go to the next slide. Yeah, so the advantages of the photopic negative response, you know, unlike the pattern ERG where the patient has to fixate, this is a full field kind of stimulus, so it doesn't matter if they're not refracted or fixating. And it's a direct indicator of ganglion cell function. It has a pretty decent dynamic range. This study actually followed a bunch of ocular hypertensives over time, and you're able to segregate non-converters. They just remained with ocular hypertension. Their photopic negative response, you could see noise in there, but the slope is basically 0. Whereas a patient who converted here, this is when they actually developed glaucoma, you could see that they were trending down and continued trending down. So the photopic negative response, I think, does have a role as a biomarker. Yeah. So a lot of in clinical trials, a lot of the FDA and regulators often require ERGs if you have a safety signal, and I'll go through some of that. Yeah, so this is actually, if you go, this is lifted directly from a talk Wiley Chambers gave in 2005 on how to use the ERG in safety trials, and he thinks that ERG should be used in drugs intended to affect electrophysiology, drugs that bind to melanin, drugs that cause retinal lesions. So if you have a preclinical, your drug causes retinal lesions, you should want to know, does it affect the function of the retina? Next slide. Yeah, so drugs that bind to melanin, he wants preclinical data to show that there are no histopathologic changes and no ERG changes, because those drugs are going to be hanging around in the RPE for quite a long time. Drugs which cause ERG changes in animals, it's not necessarily a no-go for the drug. It doesn't kill a drug if there are no associated histopathologic changes. But what should be done is that you should monitor ERGs in your patients in clinical trials, and to see if it translates into human. So I'm actually working on a drug right now that showed ERG changes in rabbits, which it was found, there's no dose response. It was found in the vehicle as well. So, but it certainly decreases the B wave of the dark adapted ERG. And, you know, there's never been any histopathologic changes. We're still trying to figure out what is causing the ERG change. Could have something to do with the potassium currents between the neuroretina and the Mueller cells. But anyhow, so ERG changes alone in preclinical studies may require monitoring, but are not, won't kill your drug. Unless you find, if you have retinal lesions, your drug causes retinal lesions and causes changes in the ERG, your development probably should be terminated. It was Wiley's advice. So Wiley said, you know, we're not the experts. If we're going to tell you to do the ERG, do the standard ERG. And that actually was part of the motivation for us to do ISEV extended protocols, so that we have a standard that can be more adapted to answer specific questions. Yeah, and he wants to see all the data. What's interesting here is that, you know, based on test-retest variability, the FDA has guided that any reduction greater than 40% in amplitude of ERG waveform would be of concern. So, yeah, a study I'm doing right now with the RETeval device in a phase 1 study where we saw preclinical, we set case definitions based upon amplitude or implicit time changes of the ERG based upon published test-retest data. We hope that this study is actually being done in Australia now because they didn't have the tox to do it in the United States. But when we go and apply for our IND in the United States, we'll have this finding in rabbits, which we don't find in other species, but we'll also have data on humans that show that it might not be relevant to humans. So it'll be important for the company. Yeah, so it's not found in the mini pig so far.
And what is it New Zealand?
Both. So yeah, we thought it might be, but it seems to be rabbit specific, and we don't. What's interesting, like I said, it's not part of the API. The active substance, the vehicle also causes it. So I think there's something that's used for solubility in the vehicle called poloxamer that I think is influencing the ERG. We're actually doing studies now to figure. Yeah, she knows about it. We'll talk later. Yeah, and we have a whole protocol on what to do if we find a case. So here's the Smurf vision. I was part of Pfizer at the time when this happened. FDA was all concerned. People were having vision changes. You know, was this a drug toxicity, and could it, especially in eyes that might have retinal disease, could this drug actually cause problems? What's interesting about sildenafil is that it works to dilate vessels based upon its affinity to PDE6, phosphodiesterase 6, but PDE5, which is part of the visual cycle. Am I backwards on that? Sorry, PDE6 is part of the visual cycle. PDE5 is what it inhibits that causes increased blood flow, but it also has a high affinity to PDE5, which is important. Next slide, Josh. In the visual cycle, in you know, working with cyclic GMP. So the question is, was this just a physiologic effect of its effect on PDE6, or was it a toxicity? So we did a dog study where we looked at a dose response after a single dose, and you could see the B wave shifting out in latency in a dose response manner. And if you looked at the drug concentration in the plasma versus the ERG changes, the changes in the implicit time of the B wave followed the drug concentration. So So the question is, we're having an effect on the retina. Is it normal physiology or is it a sign of toxicity? So we dosed dogs at high doses for 6 months, saw the same ERG change, washed them out of the drug, the ERG went back to normal, gave them a single dose, we got the same thing. So we showed that no, this isn't a toxicity. With long exposures, you're not getting any progression of retinal disease. It's just a physiologic change in the retina caused by its affinity to PD6, excuse me. And the FDA made them do a human study as well, and the same thing happened. So not all the ERG changes are killing drugs. There are physiologic changes. A lot of drugs that play with neurotransmitters affect the ERG and have nothing to do with toxicity. So I think in the interest of time, ERG is preserved across species. The A wave and B wave, look pretty similar across a whole bunch of species. And so in early drug discovery, it can be used to guide your dose selection. I picked a study from, what's his name? Carl Romano and his group in Alcon that used it really, really intelligently to guide their first in human. So if we go to the next slide. So they had this 5-HT1A agonist that they thought could be used to treat geographic atrophy. So they did a laser, not a laser, a blue light damage model. And what they showed was, so when you didn't have any drug on board, your ERG, was greatly reduced. And what you have here is a dose response. So with increasing drug levels, your ERG returned to normal by a certain dose. So you can choose, where's your plateau, which would be a good place to start, your clinical trials at. So you had proof of efficacy here on effect on the ERG. Go to the next slide. They then played around with timing between when the drug was given and showed, go back one. Yeah, so they showed if the drug was given greater than 72 hours or more before the light damage, it didn't have any effect. So you kind of got a clue on how long the drug was active. And they showed if you pre-treated with an antagonist, you could block the effect of the drug. So they proved that the mechanism was actually 5-HT1A agonism. So I thought it was very clever the way they used this. Unfortunately, the blue light damage model is not a good model of geographic atrophy, and they went and spent a lot of money on a trial and the drug failed. So you got to be careful on what model you choose. But it was very, very intelligent use of the ERG to guide their clinical trials, other than the model they chose. Yeah, they also did it with topical versus systemic. I think maybe in the interest of time, I'll skip over this section. But basically, this is another thing I did with actually Novartis on safety of an oncology drug. So an oncology drug was causing mild visual symptoms in patients. Someone decided to record an ERG and it was flat. And so there was a big panic. And we went through a whole bunch of things to explore the ERG effects. And I think if we keep going. So yeah, that one. So the drugs that cause toxicity, if you actually looked at their concentration in the retina versus the blood, the ones that stayed in the retina a long time after a dose were the ones that caused toxicity. So you could actually design your drug and look at the concentration in the retina and make sure that it doesn't stay around long, and you can avoid the toxicity in the retina. So in summary, I think the ERG is a valuable tool for the evaluation of efficacy and safety in clinical trials. Preclinical models can be used to explore therapeutic and toxic effects of drugs in the visual system, can be used to aid in dose selection, and can be used to understand adverse events that might be seen unexpectedly in the clinic. So, and again, the RETeval device is just, to me, opened up, the use of ERGs in multi-center trials. Thanks for your attention.
So I just want to talk, I just want to follow Mitch and just try to argue why kind of all the ERG devices, kind of the RETeval is probably the best one for your clinical studies. Okay, so just an outline. I'm going to do a little bit of scientific rationale, although Mitch covered that pretty well, and then a logistic rationale. So for the scientific rationale, RETeval device has had strong peer-reviewed scientific support on top of decades of prior research. I think the first ERG on a human was 1948, so it's been a while. For the RETeval device in particular, you can see this plot over until today of the number of peer-reviewed publications with it. It's over 100, including papers in AMD, diabetic retinopathy, glaucoma, inherited retinal diseases, and mental health issues. And the good thing about ERGs in general is that since the test generates numerical results, that makes them easy to interpret, and for RETeval in particular, it's been shown in many cases to be more sensitive than structural measures. So just a couple more words on some scientific rationales for my favorite two diseases. Diabetic retinopathy, the RETeval device has been used on more than 2,000 patients in 6 different countries, and it's been used as an aid in diagnosis in 9 publications, and is for monitoring treatment in 3 publications. So kind of a lot of scientific support for diabetic retinopathy. So if I could have kind of like one summary slide on our diabetic retinopathy. So these are risk factors for having an ocular intervention or having high risk PDR due to diabetic retinopathy. The first paper that Mitch discussed compared the RETeval device to photographs. If you look at the relative risks for who actually receives the intervention from it, relative risk was 11 for RETeval, 4 for photography. And then other papers like the ETDRS report 18 looked at other things like visual acuity, HbA1c levels, blood pressure, and heart exudates. And you can see none of these even come close to having an odds ratio or relative risk of that of the RETeval device. So the RETeval device really, was more predictive than all these other measures. So just a couple words on glaucoma. There's been kind of over 600 patients used in 4 countries in papers using the RETeval device for glaucoma, 6 for using it as an aid in diagnosis, and 3 for monitoring treatment. Just going over a couple of the trials. So this was a treatment trial for diabetic retinopathy. So these people were given an IOP lowering drug for 3 months. And if you look at the control versus treatment, you can see that, well, remarkably, the IOP lowering drug actually lowered the IOP. Yay, the drug did what it was supposed to do. No surprise there. What about kind of other measures? So if you look at the percent change in the photopic negative response, you see that there is a statistically significant change in the mean value. This is a short trial, only 3 months, but even after 3 months, you can see a population shift in the photopic negative response. The researchers also looked at central corneal thickness, mean deviations of visual fields, and retinal nerve fiber layers. None of those generated a statistically significant result. So only the PhNR was kind of sensitive enough in this short of a trial to generate a treatment effect. So this one, I do know the drug for this one. So this was another glaucoma trial. This was, these glaucoma patients were treated for 12 weeks with a high dose oral vitamin B3 nicotinamide in a crossover trial. So 49 patients, primarily open angle glaucoma. And if you look at the so if you look at the percent change in from baseline of the photopic negative response, if you look at the placebo at the 6-week point or the 12-week point, you see no change in the photopic negative response. If you look at the vitamin B3, the nicotinamide, at 6 weeks, no change. At 12 weeks, you have a statistically significant change. And they looked at visual fields, mean deviations in PSD, IOP, visual acuity, retinal nerve fiber layer thickness. None of those showed a statistically significant change. So again, short time frame, only 12 weeks, but 12 weeks was kind of long enough to get an ERG change. So that's all I wanted to say for the scientific rationale, because Mitch spoke a lot about it. I'm just going to now go on to logistics. So that's kind of a few points for logistics. You know, the first is the device is widely deployed, kind of over 2,000 devices shipped, kind of anywhere you probably want to do your clinical trial. We already have some devices there, so perhaps there's no new equipment needs to be bought. But as Mitch said, it's relatively inexpensive, and we do it on a rental basis too for clinical trials. So, you know, you don't have to buy the device. Okay, so let's, and there's easy to use. The device helps to ensure that only quality results are taken, and the results and test configurations are secured against modifications, so you don't have to worry about any GLP issues. And the device records ancillary information that's super helpful in trying to do post-test quality checks. So for easy to use, let's just do a demo. You might see that Josh is here. So Josh is going to test our it's gonna test our patient here. This is recent, and I think he did this a couple, maybe a month ago or so. So the first thing that you do is you can see here the time, so it's sped up a little bit, but not too much, right? So the first thing you can use, you put the electrodes on. You can use this barcode app to put in patient information, or you can key it in with the device. Then you just connect the electrode, and you can start running the test. So the test runs, this is a diabetic retinopathy test. It runs about 45 seconds for the eye, and then you switch to the other eye. The device, you know, during that 45 seconds recalibrates itself, so you don't make sure that the light stimulus is right. It's measuring the pupil size and the ERG.
So for patients with inherited retinal diseases, does it correct for interaction and nystagmus?
So nystagmus generally doesn't affect the pupil measurement response. So yeah, yes. And kind of, so, you know, it finished in 3 minutes and 40 seconds for your diabetic retinopathy test, and that was kind of from the beginning to the end. So nice and fast. So I'll go over this report a little bit more later, but you can see kind of things are color coded. You have photographs of the eye. This test does a pupil response and an ERG response. So that was kind of the demo on how easy the device was to use. Kind of only 3.5 minutes to do a test that tests cone function. Rod function is a different story. But then, you know, so how does the RETeval device help you generate good results? Because ideally you want good results. So the first thing is that you can use right here is the protocol that's used is right now on the DR assessment. If you run your clinical trial, you know, if your clinical trial is an XYZ01, that you make a protocol that's called XYZ01, and then they always run the right protocol. So that makes it nice. You know, the next thing that the RETeval device does is actually looks for expiration dates. So you had to like scan a barcode, which has an expiration date, to make sure that kind of there isn't any issues with kind of using expired stuff in your clinical trial. You know, the next thing it does is it tries to monitor. So is ERGs are measuring an electrical response. You got to have an electrode. Is the electrode actually connected and secure? So before the test, it will refuse to go past the screen until it monitors a good connection. And even during the test, if the electrode falls off or whatever, it'll stop and tell you that you have to kind of restart that test. It also looks for when it's connected, not only does it tell you kind of which eye you're supposed to test, but it will measure this electrode noise. And so if the waveform, if it's too noisy, it'll warn you so you can kind of not get bad results because you have a poor connection. So the connection's there, but it's just not as good as it should be. And finally, during the test, you see a live view of the image of the eye. So with the live view of the eye, you can coach the patient to look straight ahead or to have their eyes open during the test or things like that to try to make sure you get good results. And then there's a couple additional checks. You can, it automatically recalibrates the stimulus before the start of every test. So by recalibrating the stimulus, that you're ensured that you have kind of the right stimulus in. And it also can check for excessive ambient light. So switching now to kind of the GLP aspects of it, the results in the test configuration are really secured against modifications. So large equipment like our LKCU test system or other things, there's lots and lots of knobs to turn. You can do every test differently. In the RETeval device, you do every test the same. Once you set your protocol, roughly the only thing you can do is skip an eye, skip a test, or do a test twice, and that's it. So there's really not much you can do to try to mess it up. And in addition to the results, the PDF report includes the protocol that was run and what the details of every stimulus that was run. So it's all written down on the PDF report. The device generates these PDF reports and also generates these read-only raw data files. And those raw data files are super handy for clinical trials because we have this kind of special software package that can take data from a collection of these raw data files and output a CSV kind of results file that then gets kind of uploaded into the study database. So that makes it really nice. Pushing on. So what about those post-text quality results? So first, a little bit about the RETeval reports. So they look like this, the PDF. If we zoom in on just a fraction of the report, this bit here, we can talk about it. That one of the things it does is it goes ahead and measures the pupil diameter for you. Another thing it does is it shows replicates. It shows replicates overlaid on the plots and in the cursor table. Another handy thing is this photograph. So the photograph of the eye was taken right before the test step started. So it'll tell you, about patient compliance. Someone mentioned a pediatric study. If the kid looks like a tearing eye that's clamped closed that will never open and the amplitudes are low, probably a good idea. It's probably reasonable to assume that the kid never opened their eye during the test. The electrode position can be monitored. And if you don't see an electrode, maybe it was going to place too far away from the eye, so it's going to lower the amplitudes. The dilation status, you can see, well, was the patient actually dilated if they were supposed to be dilated? And you can even see if the correct eye was tested. Maybe you only treated one eye. Maybe you treated the left eye. Did they actually test the left eye? Or did they test the right eye instead? Right? You can see which eye it is. And then we also have these one-tailed color-coded reference intervals. So now I'm going to go, I've been the reader for many clinical trials, so I'm going to show you actual clinical trial results for both during training qualification runs and actual patient results. So in one trial, there was the dark adapted testing and light adapted testing. And the device not only measures how long the dark adaptation was, it measures how bright it was during the dark adaptation. So, 10 candela per meter squared, that's not very dark. So, query to the site. The light levels when the subject was dark adapted seems high. And the response was, oh, I used the flashlight lamp app on my phone to find the pupil. Whoops. Got him, right? So, but it recorded enough information to find that mistake, and then that was actually during a training qualification, so kind of no bad results were happened from that, right? You know, these is real stuff. These are real stuff. So, you know, another one, we got this results, and the device also kind of measures kind of what the noise floor was that was reported to them. So it gave the excessive noise warning. And you can see this is nice power line interference is leaking into the ERG. So the query was, you ignored the electrode noise warning. And they go, oh, I'm not supposed to ignore the warning? Correct, you're not supposed to ignore the warning. This was also during a training qualification.
So what's the solution?
You know, it's a training. Oh, so the solution is the electrode's not sticking well. So maybe for that patient, you have to actually take this electrode off and do some skin preparation. We can use like an alcohol pad or like new prep or something like that tries to, you know, improve the patient impedance. So in this one, there was a high DR assessment score. And the question is, was it really supposed to be high? And the DR assessment score looks at the ERG and the pupil response. So this is actually real trial data. And if you look at the pupil response as recorded on the report, you go, wow, that's a pretty non-responsive pupil, independent of the brightness of the light. And it seems rather big. And look, that pupil doesn't look very small. So, you know, query to the site, was the patient dilated before the test? And the answer was, oops, you're right, we looked, they were. So then you can kind of prevent this from being kind of, you know, erroneous results in there. Okay, what about this? So this is real patient data, real trial data too. So this was a longitudinal study. So the first 3 months, kind of the flicker ERG was, I don't know, about 35 milliseconds. And then, holy cow, look at that change to the flicker ERG at the 9-month point. You know, you know, is this the reportable incident? You know, should the trial be put on hold? Should you convene a big meeting? What do you do? Well, probably first thing you do is look at the waveforms. And these are 2 different brightness stimuli, and it kind of looks okay. And you know, it's, you know, 50 milliseconds, just not looking good for the home team here. Well, what about the photograph? Well, the electrode looks placed pretty good. But wait a second. There's a little icon that's supposed to be here. Well, where's the icon? Huh, how do you not get an icon? You get an icon by putting the electrode on like this. How in the world did you do that? The query, you put the electrode on upside down. So, and if you just, you know, oh, I'll get it right next time. So, you know, you rotate this around. Now it looks maybe like a little bit more like an ERT waveform. And, you know, you go ahead and flip it around. And, you know, you flip the waveform over and then you get that point. And look, at the 12-month point, everything looks good, right? In fact, this was caught by the reader. The CRO didn't even know about it, right? This was kind of stopped before it got to the point of trying to figure out if it's a reportable event or whatever. This was just, you know, an operator error. So these kind of things are kind of really nice to be able to do. Now, I mean, these types of errors are rare. They mostly happen during this operator certification process, but not always. And, you know, we do provide training videos, guides, and online training. But nevertheless, these checks are really nice to have around to catch the occasional issues that happen kind of during the trial site. So yeah, so in summary, kind of these are kind of the reasons why I think the RETeval device is kind of the essential device used in the clinical trials, you know, both from the scientific rationale standpoint and from these logistical reasons.